Good evening, everybody. Welcome to this evening's lecture. Welcome to Queen Mary, University of London. My name is Jeremy Kilburn. I'm from the Faculty of Science and Engineering, and the faculty started out a sequence of Richard Garriott lectures in 2011 partly to raise the profile of what we do here at Queen Mary, but also to provide an opportunity to talk about popular science to a wider audience than simply within the university. And so I'm delighted to see such a cross-section of people here this evening who come to hear this lecture. The Garriott lecture was actually named after Richard Garriott, who is a very good friend of Queen Mary. He's also a game designer, an astronaut, an explorer, and an entrepreneur. So he, in many ways, encapsulates many of the things that we try to achieve here at Queen Mary in the Faculty of Science and Engineering. He gave the first lecture in 2011, and the second lecture was given by the well-known mathematician Marcus de Sautoy in 2013. This evening's lecture is one of our own, and that's a real pleasure to be able to introduce someone from Queen Mary. It's Dr. Steve LeComber, who is a senior lecturer in our School of Biological and Chemical Sciences. Now, Steve came to the job at Queen Mary by a slightly unusual route. He was a journalist to start with, and that perhaps shows through in his ability to explain things in a particular way and to give public lectures. It says here that he came to Queen Mary in 1995 as a mature student, and thinking that if he was mature in 95, what he's like now. But actually, it's really not a very appropriate word for Steve, because there's one thing that captures him, it's youthful exuberance, and I think everyone who knows him would agree that that's very much characterizes Steve. He came here nonetheless. He graduated with a degree in ecology and genetics, and then stayed on for a PhD, which he obtained in 2003. And after a postdoc period as a postdoc for a research fellow, he then took a lectureship here in 2006. And he publishes on a very wide range of subjects within evolutionary biology and ecology, and that includes mathematical and computer models of molecular evolution and study of spatial patterns in biology. And all of those themes, I'm sure, will come through to one degree or another in this evening's talk. And the talk will be on his pioneering work introducing geographic profiling, which is a statistical technique originally developed in criminology to prioritize large lists of suspects in cases of serial crimes such as murder, rape, and arson, all the way through to epidemiology, showing how it can be used to identify disease sources from the addresses of infected individuals. And I think that, of course, shows through in the title, From Jack the Ripper to Malaria, Geographic Profiling in Biology. So, Steve, over to you. We very much look forward to your talk.
Thank you very much. Thank you, Jeremy, I think, and thank you to the Centre for Public Engagement at Queen Mary who've organised this. It really is a genuine pleasure to be giving the Garriott lecture. As Jeremy said, the first two speakers were Richard Garriott and Marcus de Sautoy, so an astronaut and Marcus de Sautoy. So no pressure there at all, thank you. But even apart from that, it's really, really nice to be giving a public lecture in the East End of London. And Queen Mary has a very long and very proud history of public engagement in the area, of which the Centre for Public Engagement, who, as I say, have organised this, is just one of the latest manifestations. But I have very personal reasons for being pleased to do this as well, and I just want to explain those very briefly in a moment. There we go. That's my grandfather. Every time I press the laser pointer, I get a beep, which I wasn't expecting. Never mind. That's my grandfather in his kilt in the Second World War. On the right, in the middle, is my father Peter, and me also in my kilt on the far right, although the more observant will notice that I've not got a cigarette in my hand. And the reason for including that is a year or two ago I started to look at my family history, and one of the nice things about having a name like LeComber is it's relatively easy to trace that side of things. My mother's maiden name was Smith, and frankly I haven't bothered. But I did get back to I have to count in my head here, my great-great-great-great-great-great-grandfather, whose first name I don't know, somebody LeComber, and his son Daniel. And the two I really want to focus on are my father Peter and my great-great-great-great-great-great-great-granddad Daniel, born in 1669. And this is the notice of the baptism of Daniel's son Bernard from 24th of July 1715, field Daniel LeComber et Anne sa femme par Bernard de la Cour. And the reason for flagging that up is, although that is in French, that's because they were French Huguenots. And Bernard was actually born here in Stepney. And everyone, up to me, in fact, for various complicated reasons, was born within a mile or two of where we are sitting now. And the two I want to highlight, as I say, are Daniel, who was a first-generation immigrant from France, and he got a handout from the Huguenot Charity Society in Spitalfields in 1739, when his address was listed as Angel Alley. Angel Alley is where the Anarchist bookstore and the Whitechapel Gallery are, and that's me in Angel Alley 200 odd years later. All the way up to my father, who was the first person in our family to go to university. He studied at Leicester and Purdue. He was head of physics at Dundee. He was elected a fellow of the Royal Society in 1992. Largely the difference because of the education he got in the East End of London. He was taught in Dagenham and at both primary and secondary school, and was the first person, as I say, to go to university. So for that reason alone, it's really nice to be doing some public engagement with science in the East End. What I actually want to talk about, as Jeremy said, is geographic profiling. So the loose plan for the talk is to tell you a little bit about what it is, how the model works, and how we can assess how well it's doing. Look at two cases in criminology, Jack the Ripper and Otto and Elise Hampel, before moving on to biology, in particular animal foraging, invasive species, and epidemiology. Finish by talking about some quite exciting current research, and then, as if the pressure of following Richard Garriott and Marcus de Sautoy wasn't enough, we're going to attempt a live demonstration, which gives me the exciting opportunity to completely mess things up in front of a very large audience. So, yeah, let's see how that goes. So, geographic profiling, first of all. This is a friend and long-term collaborator of mine called Kim Rossmo. He's a completely fascinating guy. A Canadian, an ex-beat cop, turned detective, turned professor of criminal justice. And Kim is the guy who introduced geographic profiling to criminology. And he's consulted for everyone, basically. He worked on the Washington sniper case, Operation Lynx, which I'll talk about a bit later, and any number of other things. And what Kim developed was a way of prioritizing basically large lists of suspects in cases of serial crime, like, as Jeremy said again, murder, rape, and arson. And to need that, this I should say is not a collaborator of mine. The giggle suggests you recognise Peter Sutcliffe, the Yorkshire Ripper. He, as many of you will know, was convicted in, I think, 1981 of 13 murders between 1975 and 1980. And that investigation generated 268,000 names and 5.5 million vehicle registration numbers. Now, even today, you can't easily DNA test 268,000 people, let alone 5.5 million. You can't easily interview 268,000 people. What you would like to do is find a way to take that list of 268,000 names and put it in some kind of order, so that Peter Sutcliffe is near the top. It doesn't have to be right at the top, just near the top. And the idea is you prioritize your investigation according to this method, and you just start at the top of the list and you work down. And you interview them, or you DNA test them, or whatever it is you're going to do. And you do that on the basis of the spatial locations of the crime sites. So that's the only actual input to the model. This is not psychological profiling. Okay? You just look at the spatial point pattern data of the crimes. You do some clever maths, which I'm going to talk about, and you produce something like this, which looks like a multicoloured mountain range on top of your study area, the map of your study area. And all you do is you start at the top of that, and anyone on your suspect list, anyone in that 268,000 name list who who is associated with the areas at the top of that surface is where you start. So in this case, with the red areas, and then you work down through the orange, the yellow, the green, and the blue. So what we're describing is a search strategy, and that's really important. We're not saying, aha, the murderer lives at 23 railway cuttings. Okay? We're saying, put the list in this order, and sooner than otherwise, you will get to the right person. This is from Operation Lynx, which was a series of sexual assaults spanning actually 1982 to 1985. It was the largest manhunt in the UK since the Yorkshire Ripper, in fact. The police logged 23,000 actions. They knocked on 14,000 doors, did more than 2,000 DNA tests, and the investigation generated more than 33,000 names. There was one attempted murder, 4 rapes, some GBHs and assaults. Some of you may even remember it. There was quite a lot of media interest at the time. It was nasty even by the standards of this kind of crime. And in fact, one of the victims had her eyes superglued to try and prevent her identifying her assailant. One of the problems was there were relatively few crimes over a very long period of time, and that makes investigation quite difficult. But in fact, from I think the 4th victim, the attacker stole a credit card. So police generated a geoprofile based on the locations of the crimes and also on the locations where the credit card was used. And they used that to prioritise this list of 33,000 names, and I'll come back to what happened in a moment. First, though, I want to tell you a little bit about how the model works. So we've got a graph here showing distance from my home. This is actually my house here. I was standing out in the street last weekend looking like a complete idiot, taking photographs up and down the street. And we've got on the y-axis the probability that I commit a crime. And what you can see from this is I'm most likely to commit a crime near my home, getting less likely as we move away in either direction. And that's essentially because I'm lazy. And most people are. Okay? If I want to go and murder someone, I'm not going to pop up to Inverness to do it because it's just too inconvenient. Okay? And however hard you try and spoof the investigation by doing that kind of thing, people do default to this. So you will tend to commit your crimes where it's convenient to you to commit your crimes. And what's going to be important later, that kind of thing will also apply to a lot of biological data. If you're a mosquito biting someone, it's probably going to be near the pool in which you emerged rather than much further away. So what we've got then, just to recap, the probability that I commit a crime as a function from distance from my home. So, the probability that I commit a crime somewhere, given that we know I live where I do. And that's the problem that we have to address first of all. We don't know where the criminal lives. If we did, we wouldn't need to mess about with any of the maths. What we actually want to know is the probability that I live somewhere, given that I commit a crime where I do. And that sounds like a relatively minor change, and it's really, really, really not. These are called conditional probabilities, and they have the form, what's the probability of A if B is true? And that, it turns out, is often very, very different from what's the probability of B if A is true. So to illustrate that, what's the probability that someone is over 6 feet tall if they're an NBA basketball player? Well, I checked. There is a roster at any one time of about 400 professional NBA basketball players. And when I checked, 5 of them were under 6 feet tall. So that comes out at a probability of 0.987 that you're over 6 feet tall if you're an NBA basketball player. What's the probability that someone is an NBA basketball player, given that they're over 6 feet tall, is definitely not the same thing. There are about 14,000 adult US males over 6 feet tall, because I checked, and not individually, and that would be going too far. And only 400 of those, of course, are professional basketball players, and that comes out at about 0.029. So the first problem we have to address is that the bit that is easy to do, what's the probability that I commit a crime somewhere, given that we know where I live, isn't what we want to know. So how can we get to what we want to know? Luckily, this chap, the Reverend Thomas Bayes, has solved the problem for us. And what this equation says is, what's the probability, sorry, it doesn't say what's the probability, it says the probability of A, if B is true, is given by the probability of B, if A is true, times the probability of A, all divided by the probability of B. Details don't matter. The point is we can turn one conditional probability into another using Bayes' rule. So our basic procedure is to do the easy bit. We calculate the probability of seeing the crimes we've seen for each site that I live in, or each site that I might live in. We use Bayes' rule to turn that round to the bit we want, which is what's the probability I live somewhere given the pattern of crimes that we see. And then all we have to do is integrate over all possible locations and all possible groupings of crimes, which gives us our second problem. There are a lot of ways of doing that. For just 100 crimes, that is how many things you would have to calculate. Okay? Now, to put that in context, that is the number of protons in the universe. So if you want to count just all of these, you have to first of all count every proton in the universe. And as if that wasn't bad enough, you have to do it 30 million times. Right? That's not good. That's for just 100 crimes. We can run our model on much bigger data sets, well over 1,000, when you would have to do this. You'd have to add up that many things. And actually, preparing this lecture, I thought, you know what, a really good way to illustrate how big that number actually is. I'll get a couple of people out of the audience, and I'll get a whiteboard, and I'll get one of them to read out the numbers while the other one copies it down. And I got my youngest daughter to help me with this, as I timed it on Sunday. I only got her to do it for a minute, or she'd have phoned child away. But I read out the numbers while she wrote them down. And I decided not to do that, because just doing that, just copying that number down, we reckon would take 25 minutes. Now that's just writing it down, that's not actually doing it. Okay? And again, just to put it into context, if we had checked 1 billion of these for every second since the universe began, we'd have done that many. So this is a really difficult problem. And the good news is we've solved that as well, thanks to two outstanding former PhD students, both of whom are sitting here, Mark at the top, doing some maths, and Bob. And the even better news is I'm not going to explain how they did it. If you want to know, I'm sure they'll be very pleased, genuinely pleased, to discuss it at the reception later. So that's what we do. We do some clever maths to get around the fact that we really can't be bothered counting that stupidly large number of things. So what does the model look like in practice? I gave a research seminar on this at Reading University, where one of my collaborators on other projects works. So I got her to send me a list of pubs that she'd rank in. So they're her crime sites. And the question, I mean, university ethics committees are a bit awkward about you actually asking people to go out and commit serial murder. So we're going to use the pubs instead. And the idea is, based on the locations of the pubs Louise drinks in, can we find Louise's house? Now, when we run the model, it does all of this at once. But just to illustrate what it does, I'm going to show you essentially one crime at a time. So the first pub on the list Louise sent me was 3 tons. That's the black dot here, so that's the first crime. Now, just to be clear, in contrast to the map I showed you earlier from Operation Links, the way we've done this, the highest point on that surface is the white area, moving down the yellow into the orange and the red. So at this point, with one crime, the model's best guess is that Louise lives somewhere near the 3 tons. The second pub on the list is the Cunning Man, over west of that. And the model's already done something clever, actually. It doesn't think Louise lives midway between the two. It's essentially telling us Louise either lives near the 3 tons, or she lives near the Cunning Man. Watch the right-hand peak as the third pub comes on. There we go. This peak has stayed where it was. She might still live near the Cunning Man. Now the model has updated what it thinks, and it thinks she either lives, she lives between the 3 tons and the Queen's Head, or near the Cunning Man. And all we're going to do is add crime after crime. The Lyndhurst, the Retreat, the Bull, Turks, Hopleaf, Cerise, Red Lion, and a whole bunch of other pubs. one is quite easy to solve by eye. Okay? Obviously, the place to look is around here. And the good news is the model is as convinced as I'm sure you all are that that's exactly where Louise lives. So this is the probability that underlies that search strategy. So what the model is saying, this one here, it's really pretty sure Louise lives here. But if she doesn't, it's in one of these three clusters here. So we can zoom in on that cluster in the centre of Reading, and there's Louise's house. So it's worked reasonably well. But the question is, how well has it worked? And the first few times I talked about this, I really struggled to explain the next part until I found a really nice way to do it kind of by accident, which is battleships. So I'm going to pick, who shall I pick on? I think I'll have to pick on the Director of the Centre for Public Engagement. He's now going to play battleships with me. He is, there go my promotion prospects, but never mind. So I have hidden the battleship somewhere, a one-square battleship, somewhere in that 10 by 10 grid. So you have to try and find it. So the question is, what square are you going to look for the battleship first?
E10.
E10 is a miss. I love the fact you're trying to analyse this.
A1.
Miss again.
B2.
B2, miss again. Right, this is rapidly going to get very boring for the audience. Thank goodness he didn't actually shout out the right answer. I did the sums, and there's just under a 6% chance that he would have fluked it, which would have completely ruined the next point. Now we're going to make it much easier for Peter, and we're going to give him a geoprofile, and I've obviously made it up so that it works. So the highest point is in white, labelled with a 1, descending down through 2, 3, 4, 5, 6, 7, 8, all the way down through the yellow and the red. So we'll let Peter have another go now, based on the geoprofile.
I'll guess E6.
Is a miss. Thank you.
That one then.
H3, miss. Shall I just do this? I3 is a miss. I2 is a miss. Nope, you've sunk my back. It's a much more efficient way of doing it, but how much more efficient? What we've done is we've looked at 5 squares out of 100, and that's how we're going to measure how effectively the model performs. If we look at 5 out of 100 squares, we've had to search 5% of the available area before we sink the battleship. Okay? So that's the hit score. And the hit score, low hit scores are good because they mean we've had to search less, we've had to expend less effort searching the area before we find the battleship, Peter Sutcliffe, the mosquitoes, whatever we happen to be looking for. In Louise's case, the hit score is 11.8%, which is good, but actually, for the geographic profiling model, we normally, in fact, expect much better. So I mentioned Operation Links before, and the sexual assaults and the credit cards. This is the surface that they produced, and it had 2 peaks in Killingbeck and Milgarth. And they used that to prioritise the investigation. They had a partial fingerprint, and the problem with partial fingerprints is the automatic fingerprint identification software can't deal with them. You have to do them manually, which is really difficult. So they used this to prioritise a manual check of the partial fingerprints. They turned up a match, and they DNA tested, and they matched it to a guy called Clive Barwell. And Barwell was, he confessed, and he was sentenced to 8 life terms, which he's still serving. He's actually also still the suspect in an unsolved murder. And without this, they might very well not have looked at him, because he had, on the face of it, a fantastic alibi, which is he was in prison. And it turned out there were undocumented day releases, which hadn't been recorded, and he was committing some of these crimes while he was out on day release. So in this case, those 2 peaks are in the top 3% of the surface, instead of, as in Louise's case, 11.8. So it's worked really, really well. And I guess the point I would make at this stage of the talk is that this stuff in criminology really does work. It's used by a lot of people who don't care about the maths, which I know has frustrated Mark and Bob on several occasions when we deal with them. But they want a practical tool. It's routinely used by the FBI, the Bureau of Alcohol, Tobacco, Firearms and Explosives, the United States Marines, the LAPD, the Metropolitan Police, Canadian Mounties. And it's used for serial murder, rape and arson. It's used for piracy of DVDs and of ships in the South China Seas and any number of things. And the point is they use it because it works. It's been applied either in real time or retrospectively to all of these highly unpleasant individuals. This is a series of serial killers with their victims in brackets after them. So Peter Sutcliffe moved during the investigation. The model finds both of his houses, 4.9%, 2.4%. Jeffrey Dahmer, down here, the cannibal, 8.7. The son of Sam, David Berkowitz, 4.7. The Boston Strangler is interesting because that's actually not great, 17.8%. But in fact, the Boston Strangler's victims varied enormously in age, social class, and ethnicity. And that's really unusual for serial killers. Serial killers usually stick to one type of victim. Now, he certainly killed at least one of them because his body was exhumed and they got a DNA match from one of the victims. But there are quite good reasons for thinking he didn't actually kill everyone that he said he did. One of the things this album seemed to want was the defence. So there's quite a good possibility he confessed to some that he didn't do just to get the notoriety. So that might explain why these are a bit higher. The Hillside Strangler was two cousins, Kenneth Bianchi and Angelo Buono. It finds both, but interestingly, the hit score for Bianchi is lower, which is better, if you recall. And the investigators believe he was the one who was driving it. He was deciding what to do and where to hunt. The Lost Killer, Jerry Brudos, 2.2%. The Night Stalker, Richard Ramirez, 13 victims, 9.8%. And Eileen Warnos, if anyone saw the film Monster, 3.8%. So it really, really works. Now, if you're giving this talk about serial killers in the East End of London, and as Jeremy says, you used to be you're an ex-journalist who knows a bit about headlines, you can't not consider The Local Boy, although we don't tend to stress it at open days. But Jack the Ripper. So 5 victims from August to November 1888. And the interesting bit that's going to matter for what I'm about to show you, and I should stress this is really, really speculative. Okay, this is just because you can't be in the East End of London talking about serial killers and not analyse this. But Elizabeth Stride and Catherine Eddowes both murdered on the same night, 30th of September 1888. I'm doing it again. Every time I talk about statistics and murder to the students, I realise I start to sound like Adam. And once I've thought it, it gets worse. So if I get a fit of the giggles, this will be why. I might have to start referring to homicide, actually. So this is the sites of those killings. And I've zoomed out a bit because, of course, we are here, up on the Mile End campus with the Royal London site down here, and the 5 victims here. This is Elizabeth Stride. This is Catherine Eddowes. When we run the model, we get a very simple surface. You probably can't see the contours, unfortunately, which aren't showing up, but the peak is obviously around here. And that's actually quite interesting, because the peak, the absolute highest point on that surface, less than 1%, well under 1%, is Flower and Dean Street. And the reason that's interesting is these two were killed, as I say, on the same night. And Elizabeth Stride is the only victim who wasn't badly mutilated, because the killer was interrupted and ran off, and was clearly unsatisfied, because later that night he killed Catherine Eddowes. And famously, if you know your ripperology, parts of her blood-stained clothing were found here, with a message chalked on the wall saying, "The Jews are the men that will not be blamed for nothing." Now, if he did live in Flower and Dean Street, and he's wandered off to kill Elizabeth Stride, and been interrupted, gone over here and killed Catherine Eddowes, this is on his way home. Now, really speculative, but interesting, because even at the time, Flower and Dean Street was suggested as a possible location. It was described as the wicked quarter mile, the foulest and most dangerous street in the whole metropolis. So this is Flower and Dean Street here. This is Thrall Street, immediately south of it. And they're still there. although Flower and Bean Street's name has disappeared, and Thrall Street now takes a bend north and goes along what used to be Flower and Bean Street. So I went down there. It's obviously changed a lot, but if you stand here at the end nearest Brick Lane and look along, this is what was Flower and Bean Street. So although it's really, really speculative, I do quite like to tell people that that's where Jack the Ripper lived. Now I can't prove it, because we don't know who Jack the Ripper was. So what we need is a case where we do know who was responsible. So we have, I don't know if anyone's read this book, Alone in Berlin. It was Radio 4's book of the week a couple of years ago. It was published in English for the first time 2-3 years ago. It was actually written in 1946, and it's a fictionalized account of a real case from the 1940s. Otto and Elise Hampel. And Otto and Elise were originally quite pro-Nazi until Elise's brother was killed in France, when they became very disillusioned and embittered by the Nazi regime. And they decided to foment rebellion very bravely by writing postcards and leaving them with anti-Nazi messages in apartment buildings around Berlin. So the postcards would say something like, "Germans awake, Hitler is killing their children, don't pay the winter fuel tax." And their idea, of course, was that people would read them, pass them around, start to talk, and unease and disquiet and unhappiness with the Nazi regime would grow. In fact, that didn't happen. The atmosphere in Berlin was just so terrified and paranoid. People didn't even destroy the cards. They handed them to the Gestapo. They were terrified of not reporting it. To the point that if you look at the times they were recorded, you can see that Otto was dropping them on his way to work in the morning. They were being handed in within the hour. On his lunch break, and then on his way home in the evening. But the Gestapo, for all their many faults, kept really, really good records. Right, I was about to show you. There we go. There's one of the postcards, and they recorded the location of every single postcard. And again, you can't read it, but we've got 2nd February 1942, Pulitzerstrasse, 12:30, one card. So Mark and Kim and I went to Berlin. We looked at all the Bundesarchiv material, and we plotted every single postcard we visited, almost every single one, and we plotted them on a modern map of Berlin. So this is our geoprofile. Again, I'm sorry, you can't really see the detailed contours, but you can see the lighter areas. And you can see, when you look at the raw probabilities, some spikes, including a really big spike here. And if we zoom in, this peak here, this little spike, is Otto and Elise's, that's Otto and Elise's apartment there. And it actually gets it within less than 0.01% of the whole search area. Enough actually to distinguish it from Otto's parents and Elise's sister, the other end of the street, in fact. So we've gone from the whole of Greater Berlin down to basically something this square. This is where Otto and Elise lived. And the house isn't there anymore, but I'll show you what it is in a minute. So we've got Otto and Elise's house, but one of the really pleasing things about this analysis is we pick up some other sources as well. We get Elise's brother, Alfred Lemmy, who lived down here. So they were travelling down to see him and dropping some postcards when they got there. They were starting to get nervous about putting postcards too near home. We get one that I think was probably dropped accidentally out at Siemens, where Otto worked. And I think it was accidentally because he tended to revisit apartments, and he never dropped anything else near Siemens again. And in fact, the Gestapo nearly caught him at that point, because they were looking for someone who lived in Wedding, which was his district of Berlin, and worked at Siemens. And then they were misled by a description of someone that someone else thought they had seen dropping postcards, and they ignored Hampel again. Even better, we got two peaks that initially we just didn't understand at all, at Schönhauser Alley in Hallerstadt. And there were no relatives of Otto or Elise associated with that. Nobody worked there, we couldn't work out what it was. Until we checked a 1940s tube map of Berlin. And actually, to get from Wedding, where Otto and Elise lived, to go and visit Alfred, you have to change at Hallerstadt. So they were getting the tube down, dropping some cards, and getting on another tube to visit Alfred. Alfred then moved. He moved to about here. They don't drop any cards there. They're again getting a little bit nervous. But now they have to change up at Schonhauser Alley. And we get a little fair piece there as well. So the model's found their house, their relatives, and it's found the tube stations they travel around with as well as Otto's work. So it's worked really, really well. But how well? There are 215 crimes, 215 locations, of which we have precise addresses for 205. And they span an area of 146 square kilometres. And the hit score is less than 0.01%. We have to search 0.1 of a square mile out of that 146 before we find Otto and Elisa's apartment. And in fact, if we look just at the first 35 crimes, we have 34 addresses. They were much closer home at this point, so the total area is only 34 square miles, but again, we only have to search 0.1. So at this point, we come to the admittedly rather uncomfortable conclusion that if Mark and Kim and I had been in charge of the Gestapo operation, we'd have caught the heroic anti-Nazis much quicker than the Gestapo did. Because, unfortunately, they were caught. Somebody saw Otto drop a postcard, and they were denounced denounced to the Gestapo. And this is Mark and Kim on the site of their house with a plaque behind them. And the plaque says, here stood the house in which Otto and Elise Hampel lived from 1934 until their arrest. The couple were executed in Plötzensee prison, 8th of April 1943. Their struggle against the inhumanities of the National Socialist regime was the inspiration for Hans Fallada's novel, Every Man Dies Alone, which is the German title. And actually, it was quite strange for us and for Kim in particular. I mean, I know Mark will remember, we went out for a meal on our last night in Berlin. And actually, it was quite a somber occasion. You know, Kim in particular wasn't used to wishing the criminals had got away. But in this case, unfortunately, they did. All of that is criminology. And I first heard about this actually in an article in New Scientist and contacted Kim. And we started to think about how to apply it to biological data. And it took us a while to get some suitable data, actually. And the first thing we did was look at some bat foraging behaviour, pipistrelle bats up in Aberdeen. And we found that if these guys were radio tracking the bats, and we could plug in as crime sites the locations where the bats were feeding and identify the roosts. So because they were radio tracking them, they knew where they were feeding and they knew the roosts. So it wasn't telling us anything we didn't know, but it was a really good test of whether the model worked. And actually, you know, in defence of blue sky science, at this point, I honestly didn't think it would. I just thought it was quite cool and it would be fun to try. And it did work. We thought, oh, okay, that's kind of nice. So we then applied it with a guy who was working in Queen Mary at the time, Nigel Rain, and we looked at bee foraging behaviour, and it worked again. And this very much mirrored Kim's experience with it in criminology. It was developed for serial murder, and it works really, really well. And they thought, well, let's try it with burglary, and it worked with burglary. And then they tried arson, and it worked with arson. And then they tried piracy, and it worked with piracy. So we started to think, okay, well, what can we really do? And unfortunately, the really cool paper on animal foraging behaviour was actually the only one I wasn't involved with, which was great white sharks, seals. I still can't believe they didn't get me on that one as well. But this is a paper that's published in the Journal of Zoology by Kim and two shark biologists. And this was looking at great white shark predation of seals off Seal Island by Cape Town in South Africa. So, if you're a seal, you're trying to get back to Seal Island, which is over here, and here's the little seal here. What you do is you come in on the surface of the water, and then as you get nearer, you dive down, and then you follow the slope up. Okay? If you're a great white shark, you're looking up at the surface, and you're going to come rocketing up here, and knock the seal out of the water and stun it. And the point is, there are some reasons you want to, if you're a great white shark, attack them here. First of all, there are more seals there, because that's where they live. So they all have to get back there at some point. So if you're a great white shark, that's a good place to attack a seal. The problem is, the seals are much more vigilant there, and also if you miss them, they don't have far to scramble until they're safe on land again. So there are other factors suggesting if you're a great white shark, you should actually attack them over here. So the seals are harder to find, but they're a bit less vigilant, and if you miss them, you can still chase them in as they try and make it safely back to Seal Island. So Kim and his colleagues, well, his colleagues recorded the data, Kim analysed it. Basically sat in boats with GPSs recording the position of seal strikes. And this is the data here. This is Seal Island here, and this little rock here called the launch pad, which the seals like. And if you do the profile, this isn't the full profile, this is the top 1%. Okay? What we've got is a really nicely defined area, which is the best place to hunt if you're a great white shark. And the really cool bit biologically about this is that they then broke it up and looked at small, medium, and large great white sharks. And actually what you can see is the large ones are hunting here, where they want to be. But they're excluding the medium ones, and definitely the small ones who are getting forced away from that optimal hunting location. Because even if you're a great white shark, a bigger great white shark is still capable of bossing you around to quite an extraordinary extent. So I thought that was a really cool paper. Mark got involved at this point and turned his attention initially to invasive species. And invasive species are a huge problem. They're probably a bigger cause of biodiversity loss than habitat destruction. And there's a huge amount of work on this. But Mark analysed 53 species that were historic invasives in the UK, from marine invertebrates up to woody trees. And for 52 of those data sets, not only did the model work, it worked better than any other model at finding where these species were spreading from. So the idea there is that in the early stages of a biological invasion, when you might actually be able to do something about it, you can plug the locations of the species into the model and use it to find out the source populations. And if they're giant hogweed, you can go and dig them up. If they're Chinese mitten crabs, you can go and hit them with a mallet, or whatever it is you do with Chinese mitten crabs. But again, the model worked. And probably now the most exciting area of application for this is disease biology, epidemiology. And that was where we went next. So these data are a few years out of date now, but the picture is broadly the same. We've got the top 10 causes of death and the number of people they kill in developing countries, and then the same for developed countries. And the yellow ones are infectious disease. And infectious disease in the developed world still kill 15% of people. It's round about half in developing countries. So the big three, HIV, AIDS, TB and malaria, kill 5.5 million people a year. And although there's a huge amount of mathematical work in epidemiology, almost all of it runs forward in time. So if you remember foot and mouth disease in the cull, for instance, all of that was running forwards in time. They're looking at where is the disease now, and they're trying to work out where it might go next as a way of protecting it and stopping the spread. Almost nobody does what we do, which is go backwards in time and say, okay, here it is now, where's it coming from? And there is, as many of you know, one famous exception, and literally one, which is the guy who invented epidemiology, which is John Snow. And this is the 1854 cholera outbreak in Soho. Now at this point, we've got no germ theory of disease, no one knows how cholera is spread. And Snow's genius was to plot the addresses of 616 people who died on a map. And he noticed that those deaths all clustered around the Broad Street water pump. He arranged to have the handle taken off the pump, and the outbreak stopped. We analysed the same data. So on a modern map, the black dots are the addresses of the cholera victims, red squares are the neighbourhood water pumps, this is the Broad Street pump. And if we zoom in, again, apologies, you can't quite see the detail. The hit score is less than 1%. This is the highest point on our entire surface. This is the pump. We basically got the wrong side of the road. Okay. That, in fact, isn't that difficult a problem. I mean, it's apparent from the map. Basically, the Broad Street pump is right in the middle. It's the only place to look. Pretty much any model you use is going to find the Broad Street pump. Much more impressive is an analysis we did of malaria. Now, one of the best ways to stop a malaria outbreak is to control the breeding sites of the mosquitoes who are spreading the parasite. Okay? And there's paper after paper in the scientific literature that says that's the best thing to do. The problem is, it's really difficult to find them. They'll breed in anything. They'll breed in ponds, puddles, drainage ditches, buckets, water tanks, bits of water collected in abandoned tyres, water, many of which aren't around for very long. So even though we know targeted intervention is good, you just can't find them. I've got a colleague who's been working on malaria in the Gambia for 20 years in an area of high malarial incidence. They've never yet found a breeding site, not once. Good news for us is this is exactly the kind of problem the geographic profile is designed to solve. So we looked at an outbreak of malaria in Cairo between 2001 and 2004. This is it here. So you can see this is a harder problem than John Snow and cholera. And what they did on the ground was they surveyed 265 square kilometres for standing water. They did this, and this is important later, in 2005. The outbreak was 2001 to 4. They did the survey in 2005. Every bit of standing water they found, they checked for mosquito larvae, and they found 59 that had mosquito larvae. Every mosquito larva they found, they genotyped to see if it was one of the 2 vector species that transmit malaria in that part of the world. And they found 8. We ran it in about 10 minutes on a laptop. And we produced this surface. So black dots are the addresses of people with malaria. Blue squares are the water sources with mosquito larvae. Well, that's all mosquito larvae. What we want to know is, does it find the vectors? The vectors are red. And they're absolutely at the top of our surface. And this is the bit I think is probably the most exciting. That map covers nearly 1,000 square kilometres. Even the experts on the ground had to survey 265 square kilometres to find those 8 breeding sites. We found them in 54. And actually, we found 7 of the 8, searching just 12. That is enormously useful. It's a massive decrease in search effort. It's a massive saving in time and money. And crucially, you can do it when the outbreak's happening. Okay? They did this, in some cases, 4 years after some of the cases. And even better than that, I think we outperformed the model. We outperformed the survey. If you look up here, this is actually around the university, around Ain Shams. There's all these cases of malaria, and the model is suggesting surfaces there with quite a high degree of probability. They never found any. There is no way mosquitoes flew far enough from the ones they did find to bite people there. So I think what's happened is we found sources that were active at the time, but they couldn't find them when they were there. That's the whole point. These things are difficult to find. So that's a really exciting area at the moment. We're doing a lot of work with Public Health England and with people working on different mosquito species in different parts of the world to try and improve this kind of targeting. That's kind of where we are now. Just to kind of wrap things up a bit, what I hope I've convinced you is that this stuff works in criminology. Okay? It wouldn't be used by people like the FBI and the LAPD and the Met and the Mounties if it didn't work. The same techniques, we've now got quite a lot of papers we've produced showing that it can apply to biological data. And personally, I think it could dramatically improve the targeting of control efforts, certainly in invasion biology, but most importantly, perhaps, in epidemiology. And currently, with another PhD student, Catherine Smith, who I think is here somewhere, but I can't see her. I know she's here. We're looking at TB in humans in London with Public Health England, and also in badgers. This is a camera trap I set in Somerset, looking at bovine TB. Now, the cull doesn't work for any number of reasons, but one of the things we think we could do, yeah, I shouldn't try and compete with a cute badger. It's not going to end well. We could potentially target the cull. And even on a very quick and dirty analysis with lots of flaws in the data and stuff, we could certainly have saved half of the badgers and still got the infected ones. Matt Strubig, former colleague who's now working at Kent, has just got a grant that I'm part of looking at human-tiger interactions, whether we can identify hotspots to focus conservation episodes. And I've got another project student, in fact, who's doing a fantastic final year project where we've got HIV data from the Chelsea and Westminster, and we can use the anonymised addresses of people with sexually transmitted infections, including HIV, to identify the clubs where those infections are transmitted, which is something public health people work really, really hard to try and do. It's very difficult and very labour intensive. And we were down there last Monday and we ran the analysis there. There are obviously lots of privacy and ethical issues. So we ran it on a computer there. We didn't take the data away. We don't know who it was. We don't actually, in this case, know which infection we were looking at. But we ran the analysis and said, look, we've got peaks here, here, and here. And the consultants went, well, that's this pub. That's this clinic. There are 4 pubs there that we know. There's a place license. Apparently you can get licenses for sex on premises, which is news to me. But we found sites of that as well. So they're now incredibly excited about using that to target screening and intervention. Right, that's the end of the talk. This is the exciting chance to mess things up again. Right, the demo. What we have done, or what Bryony has arranged, is she has got someone to pretend to be a serial killer. And again, ethical issues, etc. They have recorded where they spend money. And I think we had 11 crime sites. So I've got 11 locations where they spent money. And many of you very kindly gave us your postcodes when you booked tickets. So I think I've got about 160 suspects in the room. The question is, can I find amongst the rest of you penguins my polar bear? So this is the bit that is obviously going to go horribly, horribly wrong. So let me just try and sort this out. So you have to bear with me while I switch my screens. How do I turn off the mirroring? Here we go. Right. Okay. So this is in a statistical language called R. This is stuff Bob and Mark developed. So all I'm going to do is load, first of all, the crime sites, which we've called Garriott crimes. That's them in. We load the sources, which is Garriott suspects, which is you guys. And then we load a key so that I can look the postcodes up later and hopefully catch the real murder. So we check that's gone in. We give the model a tiny bit of info and we say, okay, where are the crimes and where are the suspects? Where are the crimes and where are the suspects? There they are. Right. Right. This is quite nice because we've got someone from Hastings, someone from Brighton, people from around Southend, Colchester, all over the place. That's just to show you the range of suspects. All right? We're not actually going to analyse all of you. Some of you are clearly innocent. Some of you much less so. So we zoom in a little bit and it starts to look a little bit more sensible. And then the one we're actually going to use is this magnification. So again, apologies, I don't know how well you can see this. Not well at all, actually. This is the Mile End Road, this is Victoria Park. The red dots are the crimes. Another lot down here, that's not a massive surprise. Somewhere over here as well. And then the blue, oh, one down here too, I haven't noticed that. And then the blue dots are the centroids of the postcodes in this area. So we're going to run the model, which will only take a moment or two, and see what it looks like, and plot the surface. So the model is doing all kinds of clever stuff at this point. It's going to start producing some dots in a moment. There we go. So the dots, we're not bothering to plot the map at this point. The dots are the crime sites, and it's changing the colours all the time because this was just for us. This isn't anything we would normally show anyone. But when it colours things the same colour, it thinks they're coming from the same source. Okay, so you can see it's very quickly deciding that the stuff in the top right has a common source. So it's going to count to 20,000, which is how many reps I asked it to do. There we go. It's giving us some really nice little diagnostic things, which I know Mark and Bob will like, but probably nobody else will care. And then we're doing some other clever stuff in the background, which is counting up to 300 down there. Right, there's the surface. So, what have we got? We've got one cluster around here, which is where we're sitting now. There's a peak here, and another one up here around Stratford. So we want to zoom in, but first we want to decide which of those peaks matters. Remember, this is a search prioritization. What's the actual probability that underlies it? There it is. So this is Mile End. A couple of minor peaks over here, but a really big one up here. So that is where we're going to look first. So I'm going to add a few more contours so we get a little bit better resolution. I'm going to zoom in. It's drawing lots and lots of contours now. So these are the innermost circle is the top 1% now. So let's look at this one. So it's just drawing them now. Okay, so again, apologies if you can't see it. There's the top 1% is here. And we've got one postcode sitting rather nicely in there and another one quite near. So now we want to have a look at the postcodes and see what it does. Okay. So the total search area of the whole map is 59 square kilometres. Our least likely suspect, CT54NT, who's that? Okay, you're clear. Oh, Susanna, fantastic. It's someone who who still works in our department. All right, you're off the hook. You're absolutely fine. What else have we got? We've actually got my postcode, which isn't me, but I suspect that's my next door one neighbour, Simon. He's also off the hook, so you're okay. What we want to do is look at the top of this list. This is, as I say, the exciting part when everyone says, no, that's not it at all. Fingers crossed. Right, there's the top 10. So, interestingly, we've got E14NS, which is Queen Mary, very high up. So we're looking for someone associated with Queen Mary. There's a whole bunch of other E1s here. E1, 4PE, 4BY, 4AF, 4NX. Very top, E15, 2FW, and there's another couple of E15s there. So that's our top 10. So where's Brian again? Brian. Who is the guilty? Have we got the guilty party?
You have.
What's the right postcode?
It's E15 2FW.
It's the top one.
Yes, it's the top one.
Who actually was a killer?
It actually was me.
Oh! You can probably expect that to come up at your next appraisal. That actually is slightly disappointing, because again, Mark and Bob will sympathise. We spend all our time telling people this won't identify the killer, right? It won't say who it's here. It will say in the top 10. But yeah, I think given the opportunities for disaster, I'm just going to accept that as one of those things and say thank you very much. And I hope you enjoyed the talk. If you've got any questions, I'll do my best to answer them. Thank you.
Actually, before we will do questions in it, but otherwise I'll forget. So I always do. Can I just thank Brian, not only for being here, but for all the work put in organising with colleagues this evening's lecture. A lot of work does go into organising this kind of event, and we'll continue fairly soon with the reception downstairs. And obviously there will be an opportunity to quiz Steve directly. But time for a few questions, I think, if there are some, which will allow the conversation to get more with us. So hands up if you'd like to ask a question.
Did you work with Professor Martin Neal? Because he does the basic networks software.
No, we haven't looked at that. There's a lot of ways we could build this in to either standard epidemiological models or other approaches like GIS and stuff. Mostly, to be honest, we've been trying to persuade people we're not completely mad. We took a deliberate decision a couple of years ago to produce a few more papers and just produce a little bit more evidence that this could work with biological data. And we're now starting to get a lot of people from public health approaching us. Because if you go to them in the middle of an outbreak and say, haha, I can find the sources for you, A, they're incredibly busy, they're in the middle of an outbreak, and B, they're a little bit sceptical. So we're very interested in pursuing that, but no, not yet. Yeah, right.
Are there any other fields you think this could be applied to?
Lots. Basically, anything where you've got point pattern data spreading out from a central source. So I'm talking to some guys at the British Museum at the moment about their coin database to see whether you can use it to locate Iron Age and Roman settlements, because there's quite good location data. is a peak right in the middle of the Pacific Ocean, which might be tricky. What you would tend to get, if there were data like that, and unfortunately I don't think, well I guess not, unfortunately, I don't really want to regret the absence of a lot of serial murders. This is a trap we kept falling into, saying, oh, there's loads of people dead, this is a great data set. Thinking, no, that's not a good thing at all. If you had a series of crimes like this in different cities, you could potentially look at the peaks identified in each. But then you'd have to do something different to try and hook up, well, hang on, they're all near the docks, or this one particular ship was at this point, or one person lived in all these. And I think in principle it might work. In practice, I don't think the information is there.